Hi and welcome to this video on genes. So the learning outcome around a gene is for you to understand that it's a sequence of nucleotides that codes for a functional protein or RNA molecule. As well as that, you need to know the difference between introns and exons and describe how those get transcribed and spliced. Okay, so a gene is just part of a chromosome that's going to code for an RNA. Now, if that RNA is an mRNA, that is translated into a protein that has some sort of function, or if that RNA instead is some sort of non-coding RNA, which doesn't code for a protein, the RNA itself might have a function. In this case, that's an example of tRNA, which we talk about when we look at protein synthesis. There's also tRNA is for transfer RNA. There's also ribosomal RNA, or rRNA, which forms part of the ribosome. So, as we saw in the Origins of Life video, there's a theory that RNA was one of the early molecules of life because RNA can have these functions. In this case, mRNA is a messenger that has information for how to build a protein, but in this instance, the RNA sequence itself folds and forms a functional unit, like this tRNA that can do work in the cell. So each chromosome will have many genes along its length. And as we know, chromosomes in humans, for example, come in pairs of homologous chromosomes. Now, one comes from the mother, one comes from the father, and we call those maternal and paternal. So each of us will have 2 copies of those chromosomes, and because those homologous chromosomes have the same genes and across the same locations, we'll have 2 copies of each gene. And so that's shown here in this diagram. These yellows represent 2 homologous chromosomes. They have genes A down to F, and in some cases, like as in the grey or the blue here, we have 2 copies, identical copies of that gene. But because one comes from your mother and one comes from your father, they might actually have different sequences. And so in this instance where we have the tan and the black, or the red and the orange, those genes, even though they're the same gene encoding for the same protein, might have a slightly different sequence, a different version of that gene. And we call those different versions alleles. And these alleles are where we get our information for Punnett squares and that classic Mendelian genetics of inheritance, depending on which copy of the gene you get from which parent. So alleles are just different versions of the same gene. Homologous chromosomes have the same genes in the same locations. Having a little bit more of a closer look then at the gene and its structure, what we find is that the sequence of DNA making up a gene actually has a couple of parts within it. There's a promoter where the RNA polymerase that's going to transcribe that gene and start the process of making an mRNA, if that's to make a protein. That promoter region is where it binds and starts to make the RNA. And as it does so, it will transcribe both exons and introns and make this RNA here that has those red and blue parts. What that is that exons are actually the part of the gene that's going to make the protein. The introns are sort of inserted segments of DNA that don't really belong with that protein. They might have been copied over into that DNA in an error. It might be some junk DNA, might be part of a different gene that has become interspliced with this one. So the introns need to be cut out to make mature RNA. So that process is called splicing. And there's a protein cluster called a spliceosome that will actually take out those introns, leaving the mature RNA. And this is the mRNA that we're used to that gets translated into an amino acid sequence that will fold into a protein. So when we look at DNA goes to RNA goes to protein, there is a step we have to do in here where introns get cut out, spliced out to make the mature mRNA. There's a video here on the splicing process that you might want to look at.